Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor. Welcome, everybody, again to another episode of our show, bringing you another really great guest today involved in research that is going to be creating a better tomorrow for so many of us down the road. Today, we have the honor of being joined by Dr. George Dodge, who is Chief Executive Officer and co-founder of Mechano-Therapeutics, which is a really interesting biotech company that was just recently spun out of University of Pennsylvania based on his work specializing in microcapsule development using a variety of really interesting proprietary microfluidics technologies for drug encapsulation, ultimately with a mission to help really revolutionize musculoskeletal health using this interesting platform, as well as enhance the delivery of various therapeutics for improving patient outcomes. Previously, Dr. Dodge recently spent time as faculty in orthopedic surgery at the Perelman School of Medicine at University of Pennsylvania, as well as serving as a director of the Philadelphia Veterans Administration Shared Instrument Corps, as well as director of their Translational Musculoskeletal Research Center at the Philadelphia Crescenz Veterans Administration Medical Center. Dr. Dodge has his undergraduate degrees in biology from Asbury College, as well as a bachelor's in biology and health science from State College of New York. Did his Ph.D. With a focus on biochemistry and immunology from McGill University. Did postgraduate postdoc work here in Philadelphia in the area of molecular and cell biology at Thomas Jefferson University, Department of Pathology and Cell Biology. And needless to say, has had a extremely distinguished career as an investigator with really this long-standing commitment to translational musculoskeletal research. And we're excited to have him because he's doing a lot of really cool stuff. Dr. George Dodge, thank you so much for taking the time to come on the show today.
Awesome to be here. Really excited to have this opportunity to share some of our new and exciting work that we've been doing and really translating discoveries that myself and my teammates at the University of Pennsylvania have brought to fruition for patients.
Absolutely. It's an amazing portfolio that you've been involved today, George. And I'd love to start off really, I guess going back about 35 years or so now, because I enjoy looking at sort of the background work of our guest. I took a look at your dissertation back in 1989 on these immunochemical studies of type 2 collagen degradation. And it's 35 years ago, but you set the stage back then, not just of this sort of important protein, this biomarker that was, really significant in cartilage pathology, but really in sort of laying the stage for the fact that, hey, in this microenvironment, which we'll be getting into as we talk further about it, there's these interesting biomolecules that do all sorts of stuff, from the destruction to the building up of the joint and everything in between. Take us back to 1989. Talk a little bit at the beginning of all this, because I think it's really the place where it all started, and sets the stage for everything you're going to be doing now.
Yeah. It's great that you focus on that. I frequently have to remind people that are much younger than me that there is literature and there's science beyond 10 years of their PubMed searches. So back as far as that, I did my dissertation with a noted scientist that was at McGill at the time at the Shriners Hospital, Robin Poole, who together with me, we did forge some of the first biomarker studies in matrix molecules, and I'm really proud of that. And that sort of led us to understand that fragments of joint materials, so collagen, as you mentioned, and other molecules that make up the extracellular matrix in particular, can be really monitored and observed as biomarkers for degradation or biosynthesis. So it's important. While there's hundreds of biomarkers, including moving into things like more imaging based, but all have their value. Some of them have left the mainstream of use, but biomarkers are really how we understand the disease progresses and how we can decide really whether a treatment is effective or ineffective, other than the patient saying, I feel better. So those are really, I'm proud of being part of those studies, and I kind of left those studies a bit behind in a sense and came to Philadelphia, which was coming back to Philadelphia where I was from McGill, and focused more on understanding the molecules that make up cartilage. So we, together with Renato Iozzo at the Jefferson Medical College in Philadelphia, we identified the rather unknown in cartilage sense and in the joint, the unknown molecule referred to as perlecan. This is essentially one of the largest proteins in the body, and it's found just about everywhere. And for many decades, it was considered everywhere except cartilage. But we did the genetic cloning for that, and were able to spearhead some studies that later showed that it was in the synovial membrane and in cartilage, and playing a major role in functioning as a mechanical sink or a reservoir, which not mechanical, rather a molecular sink or reservoir, where all these growth factors and molecules that may be pro or catabolic for cartilage homeostasis could reside and be left in the area to do either good or bad things, if you will. So that really moved me into continued my journey into arthritis and joint preservation, joint restoration, as it was an unmet need. Without going too far back into my personal life, I was really inspired by my mother and actually my grandmother, who both had severe osteoarthritis. And I always saw this as a need to really solve these problems, not only for my family members, but for the greater good, because it's an area that's really understudied in terms of as a serious disease. And this term I use seriously without being flippant. It's only recent that the FDA, for example, has considered osteoarthritis a serious disease on the order of other disorders. So this, unfortunately, has left osteoarthritis and joint preservation, for example, or regeneration, in the back of many pharmaceuticals portfolios. So we're happy to see that things are moving in that direction. So that brings me more to more recent things. I can skip a couple decades and move to where we get to this. That's the inspiration for my work. I like thinking myself as a translational researcher, because I really always looked for a connection to reality. And this is really where I ended up partnering with some really awesome people at Penn, creating Mechano-Therapeutics.
Yeah, I mean, before we get to mechanic therapeutics, because you were just highlighting something really important, especially for a non-scientist like myself and those that may be listening. Again, you mentioned sort of how pharma may have put this sort of in the back room over here just because for whatever reason. But a part of that is because it is we might not think about it as much as, say, the brain, let's say, or some complex 3D organ. But your work, and I encourage everyone that's listening and watching to sort of to go to PubMed and take a look at sort of Dr. Dodge's extensive publication record here. Your work, throughout this period leading up to mechanic therapeutics has been on all parts of what you term sort of this cartilage functional unit, this combination where you publish on the growth factors like fibroblast growth factor 18. You publish on 3D modeling of the tissues and this is a three-dimensional space like any other organ, and how the appropriate production and modeling of this tissue. And then you publish on what we'll be getting into a bit, sort of the physics of all this, that, hey, there's gravity, there's friction and stresses and other things that go on in this organ of the joint that we don't see as much in other parts of our body. Talk
Mhm.
about this for a few minutes, George, if you would, because I think it's extremely important because it's not just the complexity of what you're dealing with, but I think this is also why we always hear about, wow, this is a pretty recalcitrant tissue in terms of how hard it is to get things to happen in it if you're only looking sort of one dimensionally. You've done a
Sure.
lot more. Talk about this.
Right. And that's really one of the fundamental problems in studying joint tissues, in particular cartilage, because cartilage fundamentally is unique in the sense of it's avascular. So there's not a direct vascular system that infiltrates cartilage. It's really it's kind of should be thought of as more of um, a group of cells within an extracellular matrix that need to really take care of themselves. Most of the nutrition and influence is coming from the outside, either from subchondral bone, so bone marrow and bone marrow influences, which would be beneath cartilage. And then outside of the cartilage itself would be the synovial space where there's synovial fluid. This is what we see when we have an injury, an effusion of fluid that can be sometimes quite large and painful, removed. That is full of these catabolic molecules that we try to control. So it's very indifferent to regeneration by normal mechanisms because of this lack of blood supply, which usually brings in cells that are more regenerative. Now, in more recent years, we understand that there are cells within the different layers of cartilage that have potential to regenerate and can have can be more like stem cells in that they're pluripotent. They can become cells that would regenerate the tissue. But unfortunately, we don't know enough about how those cells are controlled. So we kind of are stalled at still having a rather solid material that is very functional to the joint, but rather difficult to intervene. So we do everything from the ultimate, which is joint replacement, down to replacing cartilage in the new sense of what we try to do in the community is look for biomaterials or somehow to regenerate the residual cartilage that's in the joint. So you're quite right that the cartilage itself is fundamentally its own worst enemy, I guess I would say. We know certain things go through it, which are very interesting in terms of physical properties, because cartilage is very highly charged, and so it resists a lot of things coming into it through diffusion. So this is also a mechanism by which now drugs are discovered and tagged with charged moieties that can make the drug go into the cartilage deeper. That's what I feel is a very promising, competitor to our microcapsule drug delivery. So there's a challenge here, but on the other hand, the joint is a unit. The joint isn't. You can do something to the joint that you don't have to mess around with the heart or the brain, whereas it's not systemic. And what we try to do in the new era of drug delivery is really try to find ways to not do systemic drug delivery and deliver drugs where they're needed, when they're needed. So yeah, the cartilage is the root of all of our focus, and yes, a little bit of its own evil self.
Got it. Perfect. I just wanted to lay the stage for that to give the folks listening, watching a little understanding of how this area, it's a tough one. It has been a tough one now, but clearly, you've brought along new thinking, and I think maybe it was the January 2021 paper, Mechano-activated biomolecular release in regenerating load-bearing tissue microenvironments, where you first describe, I think, some of your work with these mechanically activated microcapsules. And again, you lay the stage here that, look, we could try to inject things in there, whether it's drugs, whether it's stem cells, but there's so many things going on in terms of how, as you mentioned, the blood flow, how drugs get cleared, how, ultimately, how you synchronize some of the things we were just talking about in terms of the factors, the cells, what have you. Talk about how you came up with the idea for the MAMCs, the mechanically activated microcapsules.
Sure. So this is the collaborative work between Dae-Yoon Lee, who's a chemical engineer, Robert Malk, who's a biomechanical engineer, in sharing the position in orthopedic surgery, and Bhavana Mohanraj, who at the time was a graduate student who did a large part of this work that you're referring to in that paper. So the four of us are its inventors, its co-founders, and really, I guess I would give credit to Dae-Yoon Lee, who had already been inspired by creating microspheres or bubbles, and these bubbles, which have use in different areas, not particularly medical, but in other areas of science, and that these microcapsules, we call them MAMCs for mechano-activated microcapsule, and these MAMCs actually can have a shell that's really can be manipulated to be harder or less hard. So in terms of it having a characteristic that is can really be used as a drug delivery. So we thought of it as like we were inspired by what he had already described and realized that it could be made in a form that would be ruptured when there's a force that's equal to or physiologic or super physiologic. So these microcapsules can have a thicker or thinner wall, and they can be like deliver a payload, a drug of any kind, earlier in a disease process, maybe when there's normal ambulation or motion, or when there's some prescriptive physical therapy, for example, post-operative physical therapy, you would say not be weight bearing. So there'd be a different microcapsule that would rupture and perhaps give you an analgesic and an anti-inflammatory. So there's a collection of microcapsules that we realized very early that we could create that would deliver things that would have a distinct use in the environment of the joint.
And take that a little further now into the clearly you outline, okay, it's targeted, obviously controlled release, it's dose sparing and can limit. What is the development path at this point? Because I know I saw some animal studies in there. Obviously, these are early days of the company, but talk a little bit about sort of what's coming up in terms of the development of of this? What indications you're looking at? Take us down that path. I know it's early days, but still, really, it's a
Sure.
really exciting tool.
Yeah, let me, since you gave me liberty to do this, I'm going to back
Sure.
up just a bit. What we are in those early days, so we've been around 2 or 3 years as a functioning company. I think we had really recognized the importance of the microcapsule and how it could be used in medicine. And we first wanted to understand whether you could encapsulate something in this device through this device. This is a microfluidic device, which I could, but we don't have the images up for people to see. But it's basically a microfluidic device that has fluids going in opposite directions, and the internal fluid can contain anything you want to put in it, and the outer forces the capsule to be formed, and then post fabrication modifications can make that harder or softer. And what we really understood early on, which we were concerned about as we wanted to translate this into practice, was what can we put in it that could actually still be active. So that set the stage for where we therapeutically saw this as having great potential. And those things that we put in it, say, for example, anti-inflammatories like a drug called anakinra or IL-1RA, which inhibits inflammation, which is on the market. We could put this in it, know how much we put in the microcapsule, and look at it afterwards and actually understand that it still comes out at the same concentration and the same effectiveness. And we prove that in models and as you said, 3D constructs and things that we understood were very important to mimic cartilage and cartilage environments. And then we realized that we could also put this microcapsule into fabrics or biomaterials, which are later studies, where the microcapsule will not be only affected to rupture by crush or by compression, which you're having if you put it in the free floating joint, for example, but you also, if you have it in a fabric form or in a biomaterial where it's confined, and you apply a force, a strain, meaning you stretch it, that also can rupture the microcapsule. And it's really related to if the microcapsule is stuck to the fabric or not. So we realize that there's a wide range of applications. There's joint inflammation, there's joint analgesia, there's so pain management, there's infection control, which is a big problem in orthopedics to avoid when you have a joint replacement. And there's also areas where we have used it for wound healing, for example, where it can be placed in a fabric, much like a band-aid, where if you stretch that fabric, it releases, say, something that would enhance healing or prevent infection. Move forward a little bit and quickly is we had a second somewhat eureka moment as a team and realized that we would enhance all this by making the microcapsule adherent to it wherever we wanted it. And I think this is kind of fun, hopefully would be interesting for everyone listening. We were inspired by the chemical that's responsible for mussels, shellfish mussels, attaching themselves to rock or the bottom of your boat. And that adhesive is really strong. And that is a powerful adhesive. It's been used before. It's understood well. And a mussel attaches itself through actually collagen fibers and secreted polydopamine molecules that or derivatives thereof, which are actually really, really sticky. So we added this as a fabrication step that we post fabrication can coat the microcapsule with a polydopamine molecule, and then they stick where we want them. So this actually moves into a whole nother area, because instead of the problems that occur in most drug delivery systems and systemic delivery where it goes everywhere. And in the joint, you always have the problem where drugs come in, they have a really good function, but then they go out. They picked up by the vasculature. Our microcapsule can be put in and stuck on the inter aligning or the cartilage defect itself by this polydopamine adhesive. So we feel this is going to basically be a game changer in directing the drug to where it's needed.
Yep. I like that selfish story. By the way, I've read about that years ago in some book on biomimicry. But yeah, that's a very powerful glue. It's just interesting to see you thinking about that. I love obviously recapitulating sort of nature, but I also like what you were talking about in terms of sort of the repurposing angle here, not just repurposing for existing drugs, but again, thinking about sort of the combinations that you could ultimately put in this. And when it comes to, as we were talking earlier about the fact, when you're playing with growth factors and extracellular matrix components and so forth, it'd be interesting seeing ultimately how this sort of the poly pharmaceutical aspect to this as well. It sounds pretty cool. So no, that's all really great. What talk a little bit about, obviously I'm down the street here from you, born and raised in Philly, big fan of the Philly biotech scene. I mentioned obviously you're a spin out from Penn. Penn or PCI Ventures has been involved in helping the IP and the fundraising and all that bit. Just say a few words about sort of how things have been for you in terms of Philly biotech startup. Obviously, we're coming along like other areas of the country. We're known as Silicon Valley now, but say a few words about the company in general, how you find the ecosystem over there in terms of building a company. And you started out in the farm industry, so you have experience there as well. Talk a little bit about this part, if you would, George.
Yeah. That's a difficult question to answer in terms of because everybody's journey is different. Sure. I think Penn has been really supportive throughout the process. We had raised $5+ million in grant money from various funding agencies. That was Penn funds to the team that I've mentioned, Dae-Yoon Lee and Malk and myself. And that has been an incredible nurturing environment because of the resources, the core facilities, and so on, and processing patents and so on. With we have adhesive patent, for example, and we also are covering this in Europe and the US. So we're grateful for that journey. I think personally, my journey out of Penn to start the company has been a little rockier, and this occurs, I think, because there's when there's a mission in someone like myself and some of my teammates, outside in the company, we think of really being driven to make the company successful. We want to move quickly. We want to find the best resource and so on. Everything is a bit of a hurdle. So, for me personally, because I was a Penn employee, I needed to walk a tightrope between the two. And until recently, when I left that position and I became a full-time CEO, I can really flourish more fully in the environment of PCI, the Pennovation Center, where we currently reside. I think as we get more established, we've raised over $1 million in pre-seed money, and we're excited to have now. We're really expanded the team. We have 3 full-time employees, a couple part-time employees, and we took on an NSF award recently in June, which we're really excited about. Which, if there's time, I'll tell you the details of, which I think are really exciting and translational. So I think we're in a good spot. Your university I'll say in summary, your university, wherever you are in the country, all have rules and regulations. Try to follow them. Try to forge ahead with what you can and do the best job to really form the company and make it successful. And that's what we have done. And I'm really proud of our COO, Nick DeFilippo, who you've met, who's been instrumental in these matters, especially in the business development end. A lot of critical things to get in line to be a true business in today's world.
Yeah, absolutely. But it's nice to see you putting the team together. So that's it's and we'll definitely put the link obviously to Mechano-Therapeutics. But yeah, please tell us about the NSF grant, because I think that's
so the NSF has a program like the NIH does that most small businesses apply to, the small business awards, and there's small business technology awards. There are a variety of acronyms, but they're all related to really jumpstart in innovative ideas that I humbly accept that we're part of, and the NSF picked us up as their phase one, one of their phase one awardees. They're very engaging. They're very helpful to the company in terms of how to structure the next steps and look for phase two funding, how to commercialize products, and how to make partnerships. So it's a really engaging opportunity. What the study is itself, which we feel really strongly about, is going to move us very quickly into translation and commercial activity, is we're using what I mentioned earlier, the IL-1RA, the inhibitor of an inflammatory mediator, and we're using an equine model. And I'm quite pleased to say that the equine model is very powerful in terms of its own self is a veterinary model and a veterinary need for controlling joint disease, and which is certainly important. And it's also recognized as a closer to human model system than other very small animals. And what we've been able to do in teaming up with one of my colleagues of longstanding, Lori Goodrich at Colorado State University, which is along with Penn and Cornell and Colorado State, the three major centers for large animal orthopedics. She has a unique environment where we don't, so to speak, hurt or terminate the horse, but we leave the horse, provide for us like he or she is in a clinical trial, and we can look to control their joint inflammation, and a horse can be studied in terms of its function and its lameness as a standardized test. And we can understand, much like a human would go through, is whether this trying to dampen their inflammation of their joint with our microcapsule delivery over a soluble delivery of the same drug, whether there's equivalence or superiority in that one delivery over another, and let the horse tell us whether it's in pain or has mobility issues. The nice thing about this model, and the reason we chose it, is because the horse problem in the joint will resolve very quickly, and the horse goes back to doing what a horse does and living to do other things, that a horse does. So that's really the crux of the NSF study, but it's really a major forerunner and recognized by the FDA to be really relevant as a model for human disease.
Mm-hmm. Yeah, it gets me thinking too. I have a few buddies in the animal health domain, and veterinary orthopedics is no joke across the board. I mean, they're pretty big numbers there. So clearly, obviously the equine model is awesome, but I could see you doing dogs and cats and everything in between. That we definitely spend a lot of money on our pets, and orthopedics is a big piece of that pie. So I'm excited for you. I think that's awesome. And
thank you. It's an exciting time, I think, for the company. I think it's there are a lot of players in this world of drug delivery. I think you mentioned their own model discovery or description of our 3D model. Rob Mock has one using another a scaffold-based hyaluronic acid platform, and there's a lot of understanding and now interest in the 3D model. And what we realized that okay, now we have 3D models that recapitulate the joint tissue, cartilage. What we don't have is how to get the drugs delivered in those environments in the most effective way without really affecting other systems that require these. Inflammation, for example, is a natural process. So you only want to dampen excessive inflammation, not all the inflammation. So we've all heard enough commercials for drugs on the market. They have a lot of problems and side effects. So I think people like us trying to work in the drug delivery space are trying to target the drug to where it's needed, as I said, and hopefully partner with the right pharmaceuticals to really make that happen. I think it's about partnerships and collaboration. Could be that two systems have to work together in the end.
Absolutely. Yeah, and $40 billion a year drug delivery technology. So I mean, yeah, you're playing in a good area. And again, yeah, I personally, like the again, thinking about sort of the polypharmacy side of this as well, and how ultimately in these functional units, as you described, that you can maybe think about at some point putting 1 and 2 and 3 drugs together, and the things that can happen regenerative medicine wise. It's just really exciting. George, anything else coming up that we should know about in terms of conferences, talks you're going to be giving, anything else related to the company that's happening in 2023 as we enter 2024? Anything I missed, please take the floor.
We're going to embark in a new fundraising phase. After now this pre-seed round, we're going to release a seed round of funding, which is very exciting for the company. And we also have, fingers crossed, a DoD, Department of Defense application, because as you can imagine, I think what I haven't maybe approached is the area where the arthritis is that we really look to impact maybe first is not the long-standing, slow-acting degenerative type as much as injury-based, where an injury results in some micro damage in the joint, and then that micro damage turns into a cartilage defect. A cartilage defect turns into osteoarthritis. We want to stop that progression. So combat injuries, accidents, sport injuries, all these things are really where our microcapsule drug delivery could interject a mediator to stop those, or at least suppress that. So I think we're excited with the possibility of a big a grant from the DoD in trauma and in technology discovery with injury models and our microcapsule. So that's an area we will hopefully come back someday and tell you about how that's panned out. We're fortunate right now, I think, because of the NSF award, and I'm very grateful for that. As the team is always grateful for funding. This NSF has really of late brought us a lot of activity. So while I can't really say anything is conclusive, like we're getting a lot of reach outs and solicitations for partnering, because people understand that the NSF identifies companies with something new and novel, and we're getting into some competitive accelerator space
Yep.
because of it and things like that. So we're on a, I think we're on a good trajectory, and hopefully we'll have something moving faster to the clinic than some other things, because we're really want to just kind of end with it, that we're trying to repurpose drugs maybe initially and sustain drug delivery of something we know already works or already has
Right.
approval. So we're really always looking for partners that have shelved drugs, orphan drugs that had side effects that they couldn't manage, or
Right.
want to repurpose in a new format that we can more quickly get through the FDA and actually get new indications for everything from your joint. We have a really exciting application in disc disease and managing ruptures in the intervertebral disc, which we'll be coming out with in the next few weeks, and in dermatology as well. So I think there's a lot of things that can, that can be on the horizon. I like to think of when I have to try to explain it really hard to somebody, I say, if you want to like put our drug delivery system and say, why do we need it? I remind people that in 1950, somebody maybe said this same phrase to people that had the gelatin capsule that formulated drugs for the first time in a time
Right.
release method. So yeah, 50, 72 years later or whatever, from that day when they discovered those capsules, we're here. And I'm really happy that you gave us the opportunity to chat.
Yeah, it's an awesome story. Obviously, great science behind it. I love the fact it's Philly. Big fan of
Yeah,
Philly. And the fact you're putting together the team, getting funding. I'm rooting you on, and we'll definitely do a follow-up. I'd love to hear more about how these things all progress, because you have a lot of things out there. Again, for everybody that is going to be listening to this particular episode of our show across the various podcast networks or watching on the YouTube channel, again, you've been spending time with Dr. George Dodge, Chief Executive Officer and co-founder of Mechano-Therapeutics, doing really amazing things to innovate and revolutionize in the area of musculoskeletal health. George, again, I want to thank you for taking the time out of your schedule to come talk to us for a little while, educating us on everything you've been up to the last few decades. Obviously, thanks for what you're doing and the future of the space. And as we like to say on our show, thanks for helping to create a better tomorrow for so many people and animals out there that are going to require these tools. So
yeah,
really
totally.
great stuff. It was really great having you.
Yeah. Thank you.